This is Lab 5, which is titled Fetal Peak Dissection Part 3, Circulatory, Respiratory, and Nervous Systems by Nicole and Adeola Otuyelu. The circulatory system is responsible for the transport of a variety of substances to and between all other systems of the body. As you can see in the diagram below, nutrients are picked up in the body and delivered to cells, while wastes are picked up and taken to the kidneys for removal. This takes place through a process called systemic circulation. Oxygen and carbon dioxide are exchanged in the lungs in the pulmonary circulation circuit, which is also described in the figure below. The respiratory system is responsible for bringing oxygen to the blood and moving carbon dioxide. The different parts of the respiratory system can be seen in the diagram below, which consists of a vast system of capillaries that run through the alveoli inside the lungs. It allows for fast and efficient gas exchange. We looked at the background review of chemistry, talked about solutions, which is a homogeneous mixture of a solid, usually a solid, and solvent, usually a liquid. The most frequent choice of solvent is usually water, and solutions made are referred to as aqueous solutions. And we also looked at acids and bases, which are proton donors and proton acceptors. And the conjugate acid or base pairs of weak acids and bases form an equilibrium in solution. Weak acids, strong acids are determinant on the ability to lose hydrogen ions. The pH is defined as minus log of hydrogen ions. Similarly, pKa is defined as minus log of Ka. Given the pKa, the Henderson-Hasselbalch equation can be used to calculate the amount of the two conjugates necessary to create a buffer of a particular pH. The examples of biological buffers, which include dihydrogen phosphate ion phosphate, organic phosphates like glucose 6-phosphate ATP, and also bicarbonate system, which is a major extracellular buffer. The objectives include identifying the major veins and arteries, observing the different chambers of the heart in the pig, comparing adult sheep heart with a fetal pig heart, identifying major respiratory structures and the major structures of the brain and the eye, examining the lung, brain, and nerves through a microscope, understanding the acid-base balance in the body, and examining lung capacity. The hypothesis was the circulatory, respiratory, and nervous system are highly interlinked and function together to maintain homeostasis. Also, that the normalized number of drops of exhaled air would be higher than the normal number of exhaled air. Materials and methods was completed according to procedures titled BB2903 Lab 5 Fetal Pig Dissection Part 3 Circulatory, Respiratory, and Nervous Systems.
For the pig respiratory system, we observe inside of the pig. As you can see, the left and right lung have different divisions of lobes. The right lung is divided into 4 lobes, the left lung is divided into 2. In a human's lung, the right lung has 3 lobes and the left lung has 2 lobes. This shows one of the differences in the human versus pig anatomy. The epiglottis is used to allow air to pass through the trachea and food to pass through the esophagus. The bronchi is the largest branch in the lung and then becomes smaller, becoming bronchioles. The alveoli, which is the smallest branch, is an open sac where oxygen is picked up by the bloodstream. The pig nervous system is another section of the lab that we did. We observed the pig brain. The nervous system is divided into 2 main regions: the central nervous system, which is CNS, composed of brain and spinal cord, and the peripheral nervous system, PNS, which is composed of cranial nerves and spinal nerves emanating from the brain and spinal cord, respectively. The cerebrum is the largest portion of the brain because of the functions it interprets of sensory impulses and coordinates of voluntary movement. During the experiment, we could not use the fetal pig eye because it is too small to dissect and to observe, so a cow's eye was used instead. This is very similar to a human's eye. Observe the digestive system of the pig. Key points about the digestive system is that the system uses many organs to secrete enzymes and chemicals to help digest food. For example, amylase is an enzyme that helps digest starch. Bile is a chemical that's stored in the gallbladder and is released to break down fats. Cellulase is an enzyme that helps digest cellulose. Cellulase is located in the cecum, which is not found in humans because humans do not have a herbivore diet, so they have an appendix instead. The small intestine is the longest organ in the body. In the fetal pig, we measured the long intestine to be 317 centimeters long, while the length of the pig was 43. One of the experiments that we did was to compare room air and exhaled air. In the room air and exhaled air, we used 2 beakers filled with 50 milliliters lime water to observe after adding room air from the pipette pump and exhaled air from a straw that we would blow into. With the room air, the lime water began to get cloudy slowly, while the exhaled air, the lime water began to get cloudy quicker than the room air. The effects of exercise on CO2 production. We used phenolphthalein. So we filled 3 beakers with phenolphthalein, and phenolphthalein is a chemical that changes from pink to clear under acidic conditions, such as when carbon dioxide pulls through and produces carbonic acid. It is pink when basic, or in this case, when no carbon dioxide is in. Beaker 1 was bubbled with the pipette pump, and it took 73 seconds for it to turn from pink to clear, and 3 drops of ammonia to turn from clear to pink, because there was a small amount of CO2 produced from the pipette pump. Beaker 2 was bubbled with exhaled air, and Beaker 3 bubbled with exhaled air after 2 minutes of exercise. For Beakers 2 and 3, exhaled air was blown into the beaker from a straw for 30 seconds. Drops of ammonia were added to observe the amount of carbon dioxide present in the beakers. It took 7 drops for Beaker 2 to turn from clear to pink, and 13 drops for Beaker 3 to turn from clear to pink. We had to normalize the numbers of drops, so to find the normalized number of drops for Beakers 1 and 2, we used the equation that's shown above. The normalized number of drops for Beaker 1 that has exhaled air is 41, which is higher than the normalized number of drops for Beaker 2 that had exhaled air after exercise, which was 32. This does not support our hypothesis because we expected the exhaled air to have more CO2 than exhaled air. We expected exercise air to have more CO2 than exhaled air because after you exercise, you breathe faster and heavier than at rest, producing more CO2. It is possible that there was a human error that occurred during this experiment because the member who volunteered for the experiment probably did not breathe at the same rate for Beaker 2 and 3. The amount of carbon dioxide was greater in Beaker 1 than the amount of carbon dioxide in Beaker 2. To measure the vital capacity, we use the spirometer, and the vital capacity measures the amount of air we can intake into our lungs. Each member of the group used a spirometer to measure the vital capacity. 3 trials were performed for each member to the average. Member 1's average was 3.7, and member 2's average was 3.6.
And this shows the graph of the normalized number of drops that turned phenolphthalein pink, and the lowest values were for room air. Size air actually got the highest value and peaked at 2 different points in the graph, as shown. This is a graph of the vital capacity of the section, which is also referred to as the capacity of the lungs. This shows the table for ANOVA of the section and the normalized number of drops and vital capacity. Since the p-value is 4.818 times 10 raised to the power minus 5, then we have to reject the null hypothesis.
Here's a graph of the normalized number of drops of the entire class. I know it is difficult to see, but you can definitely see that the room air is a lot lower in the normalization number than exhaled air and exercise air. And you can see that exercise air peaks at most of the group numbers. Here's a graph of the vital capacity of the entire class. This is the average of each member from each group of the entire class. And this is the ANOVA of the entire class of the entire class. As you can see, the p-value was 1.02 times 10 to the negative 28, which is a very small number, so we accept our null hypothesis. And here's our works cited page. Thank you.